There are many aspects of conserving seeds that involve some high-tech methods. We're working, for instance, on cryopreservation, which uses liquid nitrogen and stores tissue at minus 196 degrees. These kinds of techniques need a lot of work, many decades in the case of crops. And by working together, we're able to learn from each other and work to a higher standard.
Create a cryo network globally of all interested parties by the end of 2020.
That this is just not one group, one unit doing this. There's a lot of people involved in terms of conservation, pre-breeding, evaluation, multiplication, and testing.
CGIAR are in partnership with Crop Trust in a gene bank platform that aims to provide crop diversity to the world quicker, more efficiently, with more data, and accelerate the flow and the use of diversity for improving crops, to tackle climate change, to tackle poor nutrition, to tackle all the challenges that agriculture faces and development faces. At the same time, it aims to provide a safe haven where diversity can be stored for generations so that our children can use it and our children's children in the future.
Greetings, everyone. I'm Yanni Van Veen, and on behalf of the GeneBank platform and the Crop Trust, thank you for joining us for today's Grow webinar. It's my pleasure to introduce our speaker today, Dr. Ruaraidh Sackville Hamilton. He has worked on plant genetic resources for his whole career, from 1974 as a research assistant characterizing a collection of common bean at the University of Cambridge to 2018 as head of the Genetic Resource Center at the International Rice Research Institute. Within that broad area, he has worked in diverse disciplines, always with the aim of improving the conservation and use of genetic resources. In particular, he's worked on improving gene bank management and data management, improving gene bank breeder interactions, and dealing with law and policy of relevance to genetic resources. Since retiring, he has continued to work through consultancies, which includes helping revision the CGIAR gene banks in line with the ongoing One CGIAR change process. This month's Grow title is The Future of Gene Banks: How Far Should the CGIAR Dare to Go? Welcome, Rory.
Hello, everyone. Good morning, good afternoon, good evening. A special Maganagabi to my friends in Erie. And I'm hoping some of you in Erie will be able to identify this bird scarer who's here on the field of the regeneration plots at Erie. I have to say I was a bit reluctant to talk today because I'm very conscious that the future of gene banks is you, not me. But Yanni persuaded me to talk about an attempt we went through last year with the Crop Diversity Trust to think about the future of gene banks. So what I'm going to talk about today is very much about what we did last year with the Crop Trust. This is the outline of the talk. I'm going to start with some background on the needs and opportunities for genetic resources. I'm assuming that you all know the general features of climate change and human population and so on, and the importance of gene banks for that. So I'm not going to deal with those. I'm going to address much more specifically needs and opportunities that are particular to genetic resources. And then moving on to a fairly futuristic look at conserving and delivering the right resources, conserving and delivering resources in the right way. And finally, what will the CGIAR gene banks do? And that last section I put with a question mark because that's very much for the rest of you to decide, not for me to decide. First part of the introduction I wanted to get onto was this co-evolution of technology and policy that Carolina Roa and others published in 2016, so it's a few years out of date. But I think it makes a general point about how technology has changed and policy has changed alongside it. And what's important about this is not going to end. Some people think that as soon as we resolve the current DSI issue with CBD and the treaty, that might be the end of policy issues. that's not going to happen. The technology is going to evolve, and the policy is therefore going to evolve. This is not specific to genetic resources. This has happened ever since books were first invented and people worried about the health consequences of reading. So that's going to go on forever. And we'll have to keep that in mind. As an example, this struck me last year of something that's come up since that publication. People have often felt it's quite easy. If you've got two varieties that differ in phenotype, it's relatively easy to do the genetic analysis to understand the differences between them. Much more difficult to go in the other direction from knowing the difference in their genetic makeup to predicting what their different phenotypes will be. And this that came out in Nature late last year is a fairly major step forwards in that, where now just by knowing the sequence of amino acids in the protein, you can now begin to determine the 3D shapes of proteins, which is an essential part of working out what those proteins are going to do. So this is really important example of how our technology is moving on to make us able to do more and more than we have been able to do with Genebanks. I want also to deal with the issue of the immediate impact against long-term sustainability, which has been a huge issue for the CGIAR Genebanks, especially really since the beginning of the CGIAR, because we've had the immediate impact being brought by the breeders and the agronomists, and the long-term sustainability being brought by the Genebanks. Getting the right balance there has been a challenge, shall we say, ever since the start. Always there's temptation for new DGs to come along and think they can improve the impact on poverty by taking money out of the Genebanks and putting it into breeding and agronomy. And we have to get that right balance. Those of you that have been around for a while will know that the Global Crop Diversity Trust was created precisely to reduce that conflict by providing the funding that's for essential Genebank operations and taking away that temptation from the DGs. But that was probably a bit naive. We know very clearly now it was not adequate. We need to address not just funding, but creating synergies between the Genebanks and the breeders and the agronomists. The other important feature I want to bring out is this manual from the CGIAR Excellence in Breeding project on optimizing breeding schemes. And it's probably worth quoting it in full. It is a common practice for breeding programs to make wide crosses based on the belief that genetic diversity needs to be expanded, rather than build an understanding of quantitative genetic principles or knowledge of how diversity changes in the breeding program. As a result, the allele frequency of important traits can be negatively impacted. I think that's a really important sentence, two sentences, for various reasons. One is it's highlighting this long-held discrepancy between a community that thinks it's obvious that you need a lot of diversity to reduce genetic vulnerability, which tends to be the Genebank community, and a community that sees, no, there's no intrinsic need for diversity. You only want new variants that you know are going to improve your varieties. It's a difference in attitude about how to deal with the unknown, diversity that you don't know what it does. And on the one hand, from a systems perspective, you can say it's obvious, it's going to increase sustainability. On the other hand, from a breeder's perspective, you need to work much more specifically on what you know is actually going to be better than what we have. And that's not saying the breeders are not going to take materials from the Genebanks, but it is saying that they're going to be much more careful about deciding what they take from the Genebanks, and it's realistic to do so, but it's from that one perspective that they're going to deal with diversity that they understand, not with diversity that they don't understand. I've included also this rate of genetic gain, the so-called breeder's equation, that is published in the same manual, because I think it's very enlightening. The Excellence in Breeding program focuses very much on the intensity selection eye, the accuracy of phenotyping the trait of interest, R, the length of the breeding cycle, L, and focuses very little on genetic variance. And of course, the genetic variance is the bit where the gene banks come in. The gene banks need to come in when this sigma squared A is the limiting factor in breeders' progress. And we don't really still have a way of deciding when breeders should come to the gene banks to bring in more diversity. And the other aspect of this is the A, the additive genetic variance. There's nothing at all there on the non-additive genetic variance, and yet that's a large part of what the gene banks can provide, where it's all that hidden variation that we have in the gene banks that we don't see just by phenotyping accessions. We only see when you recombine them with into different gene combinations, where all the gene interactions come into play and different phenotypes appear. So that's really outside the scope of excellence in breeding, getting at that non-additive genetic variation. And this is, I was tempted to start with this slide because I think this is really stunning slide. For me, this is based on a data set provided to me by Ken McNally's team in Erie from the sequencing of 3,000 rice genomes, where they simply listed the 50,000 more or less gene models that they had from those 3,000 rice genomes. And then for each gene model, the number of functionally distinct variants at each gene. And putting it together through this very basic high school level maths, you come up with a figure of 10 to the power of 37,000 potentially distinct phenotypes, which is a phenomenally large number. I keep hoping someone will tell me I've got my maths wrong, but I've checked it again and again, and I'm pretty sure it's right. We really need to put this into context. 10 to the 37,000, there's only 10 to the 17 seconds until the universe ends. There's very little time left before the universe ends compared with the amount of variation we have in rice. You take the diameter of the universe divided by the diameter of the smallest known subatomic particle, that's about 10 to the 45, still tiny compared with the number of phenotypic combinations. And the number of the atoms in the universe, about 10 to the 82, really tiny. If you look then, 10 to the 37,000 is approximately the number of atoms in 10 to the 37,000 minus 82 universes. It's essentially an infinite large number. You divide 10 to the 37,000 by 10 to the 82, and you get a number that's identical to the first 10 to the 37,000 more or less significant digits. Of course, most of those will be lethal, but let's suppose that 99.9999% of them are lethal, and we're still left with more or less 10 to the 37,000 distinct viable haplotypes. So it is really a huge amount of diversity, with really important implications for the gene banks. First of which is we have conserved 0% of the potential diversity of current crops. It's a question that's often asked of the gene banks, how much diversity have you conserved? Not many people are expecting the answer 0%. It's obviously not even worth trying to do better. On the other hand, with the right technology, it's perfectly achievable to conserve and document the function of all the functional variants of all genes. And I think that's what we need to look towards as the gene bank's essential long-term task in the future is doing precisely this. This is what we really need the gene banks for. And in contrast, exploring the 10 to 37,000 combinations of functional variants is basically an infinite opportunity for invention until the end of time. The concept of static conservation of genomes from the 1960s is really not appropriate. That's the basis on which the CGIR gene banks were first created, to prevent that genetic erosion from the Green Revolution. That's has to be an outdated way of thinking. But we do need to discuss, to think about very carefully the gene bank's role in accelerating evolution for future climate change and population. So we should step back a bit, I think, and think, what are the gene banks for? Clearly, they are for conserving and facilitating use of diversity for mandate species, but just what does that mean? Does it mean conserving and facilitating use of the diversity lost in the Green Revolution? And I would say, no, that's not really what we should be looking at now. There is an alternative, a subset of that, heritage varieties. Many countries value their own heritage varieties in quite a serious way. That's a difficult question for the CGIAR, because conserving national heritage is normally considered responsibility of the nations, not of the CGIAR. So it's a questionable priority for CGIAR gene banks, but we may need to be in there for helping countries that are unable to conserve their own heritage varieties. So I'd put that in orange here. And then come on to a third one, conserving the functional variants of genes that are not in breeders' working collections. I think this is clearly a very important objective in the future, along with conserving genomes of immediate interest to breeders, although this is not so clear how gene banks will interact with breeders and other researchers in this. It has to happen, but this is less clear what gene banks will do here than it is for the third one. So with that introduction, I would move on to looking 30 years from now. Let's imagine what it would be like conserving and delivering the right resources in 30 years from now. And I know the future is not predictable. The future will bring challenges and opportunities that we can't even dream about now. But just let's have the luxury of dreaming and thinking what might happen. What I'd like to see happen is that we have known and catalogued all variants of all genes. And by that, I mean not just the indels and the SNPs and coding regions, which is the basis of that figure I calculated from the rice genomes, but also the epigenetic variants, which we really haven't got to grips with at all, and the functional variants in non-coding regions, which we also haven't got to grips with at all. And I think we're really a long way off from even knowing how to deal with the functional significance of these non-coding regions. And importantly, that we need to know this not just for the gene bank accessions, but also for other known varieties, because we need to know what breeders don't have. In order to help them, we've got to find out what gene banks have that they don't have. I would hope that most functional gene banks could be annotated with a function so that we can actually make some sensible decisions. But I doubt that will happen with all variants, certainly not in all gene combinations. Technology, I can't begin to guess, of course. Things are changing every year. And so all I can put is it'll be the son of whole genome sequencing, genotyping by sequencing. Who knows what will come along in the next 2 to 10 years, but one way or another, we will get all the variants known and catalogued. With that information, we can then put together accession-specific values for conservation and use. And I'm talking about 3 different ways of assessing value. The future value, which will be quantified in terms of the number of functional variants in an accession that are not present in breeders' materials, weighted by rarity in the gene banks. So if an accession has a lot of genes that are absent from breeders' materials and absent from other accessions in the same gene bank and in other gene banks, that's going to be enormously valuable for future conservation. And this needs to be conserved long-term, regardless of value for current breeding. It may be completely useless for current breeding objectives, but because we don't know what future breeding objectives are going to be, this becomes really important to conserve these for the future. So this is really the future component of gene banks. We can also estimate a current value, which these be referring to basically pre-breeding materials with important functional variants that are in usable genetic backgrounds. And these are the ones where we'd be needing to promote use. And if they're not used, then they don't have current value by definition of the fact that they're not used, so we can discard them. And then the third one, the heritage value that I alluded to before, that's defined not by CGIAR gene banks, but defined by the countries that provided them, has to be from a country that considers the variety part of its heritage. And I suggest it needs the CGIAR to provide security. This is really has to be a joint decision between the CGIAR gene banks and national gene banks and national programs to decide the importance of conserving these in the CGIAR gene banks. With those values, accession values, we can then set about optimizing the composition of the collections. And what I would envision is that we would have quite small and quite stable conservation-oriented collections that have high future value, with relatively much larger and more dynamic use-oriented collections where the accessions have high current value. And doing this means there's going to be an ongoing process all the time of acquiring new accessions that have high future current or heritage value, and even creating new accessions that will have increased value or current value. That's likely, I would think, to become an increasingly important part of making collections more efficient and effective, is actually raising value by creating new combinations. And of course, disposing of accessions that have low future current and heritage value. And this would need to be coordinated among gene banks, of course. It's not something you can do gene by gene bank. And finally, for the 30-year vision, we need to have delivery well matched to users' needs. I would say we can expect delivery to be well matched to users' needs in a way that we just cannot achieve today. We do have to keep in mind that the users will continue to be a wide range from growers who are interested in exploring accessions for use on farm, through the researchers that explore diversity and value in the gene bank, pre-breeders and breeders who do the actual creation of novel varieties. There might be a change in balance between those users, but those four categories will remain. And to match their needs, of course, we have to have a system for monitoring their changing needs. What I expect would be developed much more over the next 30 years would be algorithms that we don't have at all now that convert users' specifications of their own needs into, if I can invent the word idea genomes, you probably all know the word idiotype. And the concept of idiotype breeding, that needs to be replaced, I think, with idiogenome and idiogenome breeding. So that would need to be together with algorithms that would optimize the breeding pathway from current material to create these ideal genomes. So that's conserving the right resources. Let's do a similar future imagining exercise in terms of conserving and delivering the resources the right way. And here, first thing I put is we would expect policy to be in harmony with technology 20 years from now. And that's fairly deliberate. I don't imagine we will ever see policy fully in harmony with technology at the time, because policy is responsive to technology. And the technology that's developed 20 to 30 years ago into the future, it will probably be too soon for policy to have caught up with it. We will, of course, we'll have to continue to make sure that acquisition, conservation, delivery, and use remains compliant with all applicable regulations. I expect these will continue to be the Plant Treaty, the CBD and its protocols, and the International Plant Protection Convention. But who knows what future agreements will come up, whether these will be independent of the other three, or whether there will be protocols within the other three, I think depends largely on their success in producing policy that's in harmony with current technology developments. It will remain, I think, very important for the gene bank community, well, really the whole CGIAR, to ensure ongoing engagement with these international fora for two reasons. One is to ensure that we continue to comply, so that we are not bashed for doing things wrongly. But the other very important one is to devise and offer technically effective and efficient solutions. I think there's a case in point right at the moment with the issue of DSI, the digital sequence information, where the plant treaty and the CBD seem to be just turning round and round in circles, not knowing where to go, because there's no obvious solution that's workable on the table at the moment. And if the CGIAR can come up with effective and efficient solution for that, that would be the way in which these international fora could start to make progress again. I don't yet see that happening, but we need to do that sort of thing. And to devise and offer them from the CGIAR is good not only for the international fora, but obviously good for the CGIAR to make sure that policy fits what the CGIAR expects. Management targets, we've got to have, again, unchanged. Accessions will have to remain viable, healthy, genetically true to type, protected against risks of loss and available to use. But we can now, or then in the future, be able to tailor accession management to their specific value. So if an accession has high current value, you don't need to do viability testing for that, because if it's not used, you no longer need it. It doesn't matter if it loses viability. As long as it's used, it will be multiplied and it will remain viable. So there's some efficiencies we can gain through the accession valuation scheme. We do, of course, need to do a lot more to introduce 21st century management materials and process management. There are things like enhancing quality, increasing throughput, reducing costs, as we can do a lot more about to achieve this through things like automated seed handling. And I've listed a few: withdrawal from and deposited in seedling, all the sorting, counting, weighing, labeling, packaging, planting, the testing for viability, health and genetic integrity. There's a lot of potential for doing this automatically. And likewise with phenotyping, genotyping, and sequencing. Of course, this needs substantial retraining of staff to operate this machinery to achieve higher throughput. And really, automation is only workable when you do have high throughput. If you've got Genebanks doing not very much, then there's not really any point in investing in automation. So that's a bit of a conditional one. How should we get there? And this is where I start to open the floor for questions, because that needs a lot of discussion. There are some areas that are highlighted in the document. And I know there's been a lot of discussion over the past several years, and that'll continue to go. The concept of smart evaluation is a very important one. But we have to increase knowledge of diversity and the genetic control of traits. But we are never going to evaluate whole collections for everything. It's just impossible. So we have to work on partial evaluation of prioritized subsets for prioritized traits, and that needs to be used to direct future evaluation. So it's a sort of an iterative progress of evaluating some and then working out what you're going to evaluate in the future as part of your gene discovery process. It will help to adapt and adopt automated and image-based phenotyping, but that's only part of it. Even with the best possible automated phenotyping, you're still never going to automate everything, so it's still got to be prioritized. Characterization, that has to continue for everything, but again, this will need to be changed, recognizing that characterization really is our basis for monitoring genetic integrity. So it's essential for every accession. There will need to be a crop-specific migration to DNA-based fingerprinting, and that means how quickly that will happen depends on the crop. There will need to be image-based phenotyping for key descriptors, like if you're sorting seed, you can have an electronic seed file. This is going to be the only way of doing high-speed seed sorting. You can't do high-speed seed sorting with DNA. And so the only way to do it at high speed automatically is through image-based phenotyping. Other phenotypic characterization traits, there's got to be some discussion on, do we need all of them or can we drop some? Genotyping and sequencing, again, that's going to be crop-specific. Migration towards gene-based methods is obviously going to have some whole genome sequencing as part of identifying what genes are, but not necessarily entirely whole genome sequencing. That's something that's probably tried as we go and modified as the technologies evolve. And then there's the issue of supporting innovation, of course, to get the synergies. This has to become an increasingly important part of what gene banks get involved in somehow, not necessarily do because there are other people doing it as well. The issue of creating breeder and research-oriented developments, creating research populations that are optimized for gene discovery and annotation. And this can include subsets for whole genome sequencing with GWAS, with genome-wide association studies, which is the first stage of helping to identify functional genes. One I would highlight is creating novel genetic resources with the purpose of maximizing creation of new combinations of genes and therefore exposing the hidden variation that's present in the gene banks. I think this is really important for the gene banks to get involved in because it's completely different from the breeder's approach to pre-breeding. The breeder's approach is to screen materials, whether they're wild relatives or land races, screen them phenotypically to look for the ones that are already showing desirable traits, and then put them into their pre-breeding programs. So their approach is just not going to expose the hidden variation in the gene banks. We need a complementary approach based on choosing parents based on their genotype rather than on their phenotype. So I think that's going to be an increasingly important part of the gene bank. Also, an increasingly important part is the digital gene banks, where recognizing that access to the information is just as important as access to the material. And it's got to be searchable information, not only on the materials in the gene banks, but on the materials elsewhere, as I said before, so you can see what's in the gene banks and not elsewhere. Obviously, there are so many systems already in place. It's going to be a distributed information system based on what all the other systems are. A really big investment is required to integrate them, and the global information system of the treaty is a start. It's an obvious starting point, but there's a long way to go from there to what we would really need to make it useful for the gene banks. Then the next steps is talking about improving each gene bank's efficiency, effective throughput, and quality, so we do things the right way. We've done a lot towards, with Yanni's guidance, we've done a lot towards improving SOPs, but there's a lot further to go. There's the question of the role of automation in these, in the different areas that I've mentioned. I've mentioned both of these as 30-year targets. We should be there, but we've got to have a way forwards to achieving these. But it's not just how each gene bank improves, it's larger scale improvements like the service provision, CGIAR, system level improvements, and global integration beyond the CGIAR system. And very little of this is going to be possible properly without the R&D that we need for improved effectiveness. I think an awful lot of people out there who are not really familiar with gene banks think that we worked out back in the 70s and 80s how to run gene banks. But we're learning more and more just how much more needs to be done, how much we don't know. And I've highlighted these 4 areas of seed longevity, cryopreservation and tissue culture, crop wild relatives, forage and trees, and phytosanitary controls. We just cannot improve gene banks unless we know how to improve them better. And this is going to require a lot of R&D. So there's going to need to be some sort of serious program in this research area. I'm not going to go through them all in detail, but picking up one or two examples, there's an area where no one has tried at all. So I picked this out because it's an area I think it's calling out for automation if you do it on a large scale. You look what happens when someone asks for seed. You go into the seed store, you take it out of the seed store, into room temperature, you've got to leave it there for a day to equilibrate. You then open it and check it, and you have to divide it into two subsamples, the bit that goes back and gets restored in the cold store, and the bit that goes out to the requester. There's a lot of steps in all of this, plus the delay and the change in temperature and the opening up and exposing to humidity, which is really inefficient and ineffective. I know a lot of people refer to the Japanese gene bank as an example where they've automated seed storage, but to my knowledge, they haven't gone this far. We need to do what they have done, but go a step further to get all of this process. If that could be done automatically, actually inside the cold store, it could be done within minutes rather than days. That's a pretty big change in the current process. So it's not going to be sensible to do that unless you're doing it in a really high volume way. Service provision. I don't want to go through each of these, but there's a whole lot of things that gene banks do that traditionally every gene bank has felt it needs to do all of these, but that's simply not the case. Not every gene bank has to do everything in-house. There are plenty of opportunities for outsourcing services and making everything more efficient. Which kind of raises the question which has been discussed a little bit last year. I don't know how far it's gone since then, especially in the concept of one CGIAR. Should we adopt a USA or India-like model where they have a centralized facility for long-term storage and medium-term storage, and then a whole set of hubs? Well, I put them as regeneration hubs and distribution hubs, which are distributed around where they're needed. If we do this, then everything can be brought together and they can be specialized. We can do everything in the central facility. It can be optimized for long-term security. We can have full testing for viability, health, genetic integrity, which would be very different from a generation hub where you're optimizing for production of high quality seed, and the distribution hub where you're optimizing for interaction with users. It's a difficult question with lots of pros and cons. One of the pros is, of course, you can put this under single management. You take it away from the risks of DGs thinking they want to take money away from gene banks and put it into breeding. You can get the benefits of scale, at least within crop groups. If you bring all crops of one group together, you can introduce automation, specialization, and harmonization of standards, because you'll now be working at a volume, a throughput level where this really makes sense. You'll find it easier to engage with international fora. But of course, if you're not careful, this can lead to a lot of international transfers for gene bank management, just internal, what should be an internal transfer from one aspect of a gene bank to another aspect of a gene bank. You really don't want to do this. We know that international transfers introduce a lot of complexities, a lot of expense, and that needs to be minimized for managing an individual gene bank. And of course, it's going to be politically very challenging, which is going to be the country or the countries that get to be the single central facility. So really a single global facility is actually not possible and desirable. So we can't go all the way towards a USA or India-like model. But we can imagine perhaps 2 or 3 centralized facilities. And that needs some serious discussion, which you may have been having already, and I'm not sure where you've got to so far. And the final one, well, the global integration. I've just put up some of the players in plant genetic resources, really just to highlight that the CGIAR gene banks are just one of a lot of stakeholders that have an interest in plant genetic resources. The other gene banks at various different levels, the immediate users, the primary immediate users of the gene banks, and the people who actually get the material out into production, farmers and local communities, and of course the people that sit over this all and deciding the rules and regulations under which we have to operate. We could draw bi-directional arrows between every pair of all of these, but obviously that makes a nonsense of this, and I'm not going to begin to go through how to connect these in any rational system. All I've done is put whether the GPA, the Global Plan of Action, and with it access and benefit sharing, because how these work together depends very much on the high level of what we're thinking globally in terms of working together and the Global Plan of Action and on benefit sharing. So that's all I want to say about that. And with that, I think I will just say thank you and hand over back to Yanni.
Thank you, Rory. That was fantastic. Nice walk through what you think the future should be. We have a few comments. A warm regards from Viviana Becerra from INIA, Chile. She says it's good to see you. Noel Anglin, great talk. I was curious if rice has 10 to the 3,000th variants. What do you think the number of variants is for outcrossers and polyploids? Also, how do you suggest genetic identity is measured for seed crops, and how much investment should be sunk into this since it's a no-end game? So first question is, what do you think about the variants for outcrosses and polyploids? Take that, if it was just not considering even outcrossing and polyploids. What is that number? What is that number?
Indeed. I would guess that Noel can answer that question much better than I could, because, and as you know, the consequence of outcrossing is that you get much more genetic variation within accessions relative to the variation between accessions. And on that basis, well, you could say you don't need to do as many accessions, but you have to find out a way for handling the variation within accessions. So you could find that the number of samples you deal with is potentially much more, unless you find a way of doing lots of individuals in a single sample. And I know that's been a large part of CIMMYT's work over the last 20 years or more. It is working out ways in which molecular methods could be applied, so you can do a single analysis of a bulk of all the seeds in an accession. So that requires methods that give you a quantitative variation in band strength, for example. So you don't just get this allele is present or absent, but this allele is present in X% of the genotypes of the accession. If you can do that, then the task probably becomes achievable in a way that it hasn't been done with rice. The approach with rice has been to purify the sample so that one accession is actually just a single genotype as far as possible, as far as you can get just by selfing, which is not 100% of the way, but it's enough of the way. It's enough for rice, but it would not be enough for maize. So it's a very different approach for outcrosses. For polyploids, also different, except in some ways for the clonal crops, the clonal polyploids like potatoes and so on, it's potentially easier because you got it true breeding through the fact that you can propagate it clonally. And that introduces great simplification compared with rice. So for rice, we've had to get 2 samples of each accession, one that's purified, and that's the one that's been sequenced, and therefore that's the one that all the phenotyping has to be done on if you're trying to do the gene discovery work. But of course, you can't get rid of the original diverse accession for other purposes. You wouldn't need that with potatoes because you can propagate them clonally as long as you use a method that can handle heterozygotes, which is actually easier than the case with maize, where you would want to bulk multiple versions. All you have to do is get a very limited numbers of alleles present making up a band. So I think the polyploid case is in some ways easier than rice, in some ways a little bit more difficult, and it's really the maize one that's quite a big challenge, unless its efforts to combine multiple accessions into a single test works well for sequencing.
Okay, Noelle has a second part to her question, and that is, how do you suggest that you measure genetic identity for seed crops? And how much investment should be sunk into this, since it seems to be a no-end game?
Yep. This is, okay, it depends on what level of genetic identity you want. I'm not talking at this stage about genetic identity for identifying duplicates. If you're talking about genetic identity for being confident that a new generation of an accession is the same as its parent generation, in this case, you have a high expectation that it is the same, unless you've made a mistake in mislabeling, unless you haven't taken adequate control of cross-pollination and seed contamination. But these are all things you can take control of through a decent SOP. Through controlling your regeneration properly, you have a very high expectation that the progeny is the same as the parent. What that means is actually you don't need a very rigorous fingerprint. You could have a quite low density fingerprint, maybe, well, I know at Erie they've tried 10 or 20 SNPs, and I don't think that's adequate, but 50 to 100 SNPs would be more than adequate to confirm that the progeny of an accession after regeneration is still essentially the same as the parent. Dealing with duplicates is a much more challenging task, because in that case, you don't have the prior expectation that two different samples are the same, unless perhaps you've got some passport data indicating that they are the same. And if they have got other data, again, you could accept a low level of fingerprinting density. Trying to identify duplicates when there's no prior evidence is really a much more challenging task, and that's a no-end game, and that's one of the reasons why I've always felt that trying to remove duplicates from accessions from collections is not really a high priority task compared with the other things. I haven't even put it in here because it's not a cost-effective way of going forward with gene banks, except for clonals. That's always going to be the case. So for clonal crops, that the cost of conservation is so high, and the cost of identifying duplicates is so relatively easy that it does make sense for clonal crops. But I don't think that no-end game doesn't make sense for outcrossers and inbreeders.
Okay, we have a question from Jacqueline Hughes. Rory, we generally aren't that good at influencing policy, particularly country policies. We need to focus on this aspect if we're going to make huge strides. How do you think we can, as a community of practice, influence policies at the country level?
Hi, Jackie. Nice, good question. Maybe I should ask Michael Hailwood to answer this, because I think, Jackie, you say we haven't been very effective, but actually our experience, I think, when you engage in these international fora, if you take the right approach, you can actually be quite influential. I would say probably not at the national level. If you try to influence individual governments, they will very quickly turn on you and say, your job is to help us. It's not to tell us how we should decide our policy. But at international level, when you get into these fora and they're debating issues like DSI and no one's coming up with a solution, if you put it in the way of, here's a proposal that's technically workable, I'm not telling you how to do your policy, but this is a technically workable solution. Actually, they do listen. There's been some quite good experiences where CGIAR is innovative, doing something that works, and at the international fora, they accept it. So I wouldn't be quite as negative at the international level. I would definitely agree that at the national level, when you try to interfere with policy, that's you'll never get anywhere with that level.
Okay, thank you. A question from Peter Bretting. Wonderful, incisive presentation, Rory. A question about the term functional variants. Are these equivalent to alleles, haplotypes, genotypes, traits, all of the preceding? Some? Again, many thanks for a thought-provoking talk.
Okay, thanks, Peter. Glad you're on too. Yeah, I chose the term functional variants rather deliberately to avoid all those other technical terms. It means any variant in the DNA that causes a difference, or in the chromosome, I should say, that causes a change in the phenotype. And that could be, it could be a SNP or an indel within a coding region. But there's an awful lot of variation that is not determined by that. We know that the epialleles, I think some people are calling them now, have just as much effect as real alleles. Those are functionally significant. So I would include them as a functional variant. And similarly, the regulatory regions of genes, which I don't know how much the experts know about them now, but I find them incredibly confusing. I have no idea how these regulatory regions work, how a sequence difference within a regulatory region is leading to a change in gene expression, but somehow it does. And therefore, that's a functional variant. So if you're thinking of an allele as just a sequence variation within the coding region, then I would say functional variance is much broader than that. Anything that affects phenotype. Is that adequate?
I think so, yeah. We have about 10 more questions, Rory, so I'll just plow through. From Hafid Aberkane, I hope I pronounced that right. Should the CGIAR gene bank in the future be involved in research/use of the available diversity more than focusing on conservation?
That's the big difficulty, isn't it? How much goes on conservation and how much goes on research and use. Maybe we should go back to what the Global Crop Diversity Trust was created for, which was to support the conservation full stop. And whatever funds the Crop Diversity Trust comes in for that purpose from that endowment fund, that should definitely go on conservation and should not go at all on research and use. That would be a misuse of the Crop Trust's remit. And we can't ever get away from that. That's if we get away from that, then we lose the reason for existence of the Crop Trust. Research and use has to feature. I mean, it always has featured to some extent, to differing extents depending on the gene bank. And where the balance is between what the gene bank does and what its users do is probably going to be variable and depends on who's going to provide the funding. Certainly, the gene banks have to engage with the users. And that's at the minimum, that's going to be, if the users are the ones that do the research and use, okay, the users do the research and use. And the gene banks just do the engagement, sufficient engagement to work out what the researchers and users need to do with it. That's a kind of a minimum level, which would work if the researchers and users had the breadth of interest that we think is needed for the future of gene banks. And I'm not sure that that's going to be the case, because most R&D has a very short-term focus. R&D is mostly about making sure that we can help eliminate poverty next year, the year after, or deal with climate change now, mitigate and adapt to climate change as soon as we can. But there's so much in the gene bank, that so much hidden variation, so much potential value that will never be, well, would take a very long time to be realized if we accept that minimum, that I would say we have to go beyond that, just that minimum. I took an example of the pre-breeding, where breeders of the breeders of the CGIAR are very clear. They know how to do the pre-breeding, and they're very clear that their starting point is a phenotype where you have a demonstrable, valuable trait that's in an undesirable genetic background, and the job of pre-breeding is to transfer that valuable trait into a better background, so that it can then be introduced into the breeding population. And that's a perfectly valid way forward. It's a very good and efficient way forward for the short term, but it does nothing to address this issue of all the hidden variation that's present in the gene bank. Hidden because you just don't see it when you evaluate the materials, and it only becomes apparent when you create novel crosses. And so I think we came up with the term of discovery breeding to distinguish from the pre-breeding that breeders do. Discovering breeding that reveals the hidden diversity in the gene bank. I would doubt that you're going to get breeders doing that kind of pre-breeding. And therefore that becomes a priority area for gene banks to get involved in research and use. So I don't think there's a single answer, but I would see somewhere beyond the minimum of just supplying materials to researchers and users, but a careful and ongoing engagement with users to make sure that the gene bank does what needs to be done.
Okay. Michael seeks an abbreviated answer about prioritization and practicality. And his question is as follows. You estimated the indefinite genetic variance possibility for rice, and you talked about epigenetic variants that may be also crazy, that's his word. Then comes the specificity between seed and clonal or even tree crops. So practically, how do you see the way forward in terms of prioritization when you dreamed about gene banks exploring and captured the useful parts of these infinite possibilities?
Okay, well, I can give
a recipe. 1, 2, 3. That's what he wants.
No, very brief answer is that's your decision. I'm out of it. I don't know how to get there. This is you got to decide what's the most achievable. And even since I left IRRI, there have been advances in technologies. It doesn't make sense for me to prioritize on your behalf. I'm sorry, I'm not going to do that.
Okay, that was quick enough. All right. There's a comment from Hans. Research leaders need to ensure that gene bank managers and practicing breeders work together to identify the useful and usable diversity in the gene banks by using the product concept, idiotypes or idiogenomes, for the different growing conditions for a crop. So he basically put that thought out there, and I think you have mentioned a thing or two. From Mr. Siddiqui, what support is there, or there's one above, could CGIAR systems support the underdeveloped gene bank to identify the high value accessions to conserve at all costs? In other words, what help can come from CG systems to national banks or underdeveloped banks?
Well, I think that's always been one of the priorities for the CGIAR gene banks, is to help, especially the underdeveloped gene banks, the gene banks in underdeveloped countries. We know they have a lot of difficulties in just basically maintaining their accessions, and it's covered the whole spectrum of activities, from data management to regeneration to characterization. There have been a lot of training programs, and obviously now that has to move on to helping them work out at a DNA level what's in their collections. And as I was saying, we can't do this just for the CGIAR gene banks. It has to be done across the world, and that includes other gene banks, and it includes the breeding materials, because this has to be rationalized at a global level. So if we don't do this for gene banks in developing countries, we're just not doing it properly. And I'm sure that will be a large part of what the Global Crop Diversity Trust gets involved in, is making sure that there's an appropriate allocation of resources to help the underdeveloped country gene banks in the way that they need to conserve what they really need to conserve.
Okay. There's an interesting question here from Prasad. At the moment, a variety is defined using DUS, meaning the UPOV, right? When would that change to DNA-based identification? Is it feasible and practical?
Yeah. You have to ask UPOV that.
Yeah.
I'd say that's just for new varieties. They don't necessarily work for the traditional varieties which we have in the gene banks, because well, DOS is distinct, uniform, and stable, and traditional varieties are generally not distinct or uniform or stable, or they might be distinct. We used to come up with the term, we should get the US out of DOS, because traditional varieties are not uniform and stable. But to get back to the question, UPOV has always resisted going to molecular methods on the grounds that they really needed, I think, something that was functionally significant. So they wanted phenotypic characteristics, but because anonymous DNA methods were 99% non-functional variants. And that using those really opens the way to allowing what are they called, essentially derived varieties, to be regarded as totally different varieties. And that's really impossible. You cannot have that in DOS. I haven't talked to DOS, to UPOV for several years. So I don't know what their current thinking is, but I think now that we can distinguish between the different categories of DNA and get functional polymorphisms that identify the real functionally distinct attributes of a variety, it's now technically possible to move into meaningful DNA-based characterization for DOS. So I would have thought UPOV should be changing at some stage in the near future, but that depends entirely on getting appropriate molecular methods for the genes that distinguish one variety from another.
Okay. There's a question from Govindar. How do you see opportunity to explore CGIAR gene banks, some yet to be characterized in 21st century? For traits that are unexplored, such as micronutrients and vitamins, you need of the hour of human and animal health. Any suggestions to gene banks and breeders? How to explore some of those characteristics that have not been explored yet, as micronutrients and vitamins, for example?
Yeah, indeed. That's a very important area. I would go back to the earlier one. Who was it that made the comment about the product profiles and working to idiotypes?
Mm-hmm.
Really, the gene banks have to be the followers in this, not the leaders. it has to be the breeders, based on ultimately what the social sciences and policymakers decide, what are going to be the breeding priorities. And as soon as micronutrients and so on become breeding priorities, then there's going to be a reason to start screening gene bank accessions for these. But the gene banks, I don't think, can proactively make their own decisions on which traits should be considered important for the breeders. Those are breeders' decisions, not gene bank decisions. I would hope they do make those decisions. I would hope we can start to assess them for micronutrients and so on, but that's not a gene bank decision.
Okay, there's a question from Charlotte, and it's actually related to another question below. How can we get to a point to confidently know all functional variants? Won't this be a journey of discovery forever? Won't there be a functional variant knowable only in future contexts?
That's probably, I don't know if Ken McNally is there, but that might be a question for his team, but because areas going on beyond the 3,000, it's clear from the 3,000 that 3,000 is not nearly enough, but you can do the sampling statistics of showing how much new variation you're discovering as you screen more and more varieties. And you'll get to a stage where you find a reduced return on investment. It's typical. The more you invest in something, initially you get a lot of return on your investment, and the rate of return on investment gradually declines as you do more and more. And that will be something we learn as we sequence more. And there will be a stage where it's decided it's just not worth screening more and more. We've done enough whole genomes, and no one's going to want to do more until perhaps the price comes down another 100-fold. And maybe in 30 years we might be sequencing whole genomes in 2 minutes with at the cost of 2 cents. Who knows? And then the rate of return on investment is it'll be just something you do as standard, just as we do now. Look at the height of every accession. We do the characterization of every accession, and the sequence will be easy. It's very much, it's an economic issue, the cost benefit of the cost of doing it versus the return on your investment.
Okay, we might take a couple more questions. The other ones are more comments. There's a open philosophical question about rice, and there's a very down-to-earth question about COVID. Which do you want first? Let's do the COVID one. You can close with your philosophy. I think that would be a better closing one. In a situation like COVID pandemic, this is from the Mr. Siddiqui, where transport and mobility is compromised, how do you see the centralized facility versus regional versus local question if we have something like COVID again, where people are grounded?
Yeah. That's I haven't had a question like that before. I'm not sure to what extent it's going to matter. I can't see it at the moment. Maybe I'm not thinking fast enough. But I mean, this central facility is never going to take the place of farmers. And if you imagine another COVID in the future, and where everyone is isolating, the farmers are going to continue growing their own crops. They'll have the diversity that they're working with at the time. The centralized facility will continue to work in an isolated fashion. So there won't be a long-term impact, I think. We can even now, I'm not aware that COVID has prevented movement of seeds across boundaries, because you can take measures to sanitize it. That's already part of the process, to sanitize. So maybe I'm missing something, but I think we can, over limited periods, operate in isolation as we have in COVID. If you can show me cases where COVID is prevented sharing materials. Yeah, that'd be interesting to know. It's an interesting question.
Yeah.
Sorry, I can't come up with a good answer.
And the last one, I think, for today, in your opinion, what should the ideal rice idiogenome have today? If you had to construct it or talk about it, what would that look like?
Well, that's a bit like the question I'm often asked is, which variety of rice do I like best?
Mm-hmm.
And the answer is always, it depends on what dish you're cooking. The idiogenome for basmati is totally different from the idiogenome for risotto rice. Is totally different from the idiogenome for Thai rice. All these different types of rice, which you cook in different ways, need to be different. And that's just about flavor. There's also the question of where you grow it. Every farmer has a different soil type in a different climate. There isn't a single idiotype, nor is there going to be a single idiogenome. The idea is that once you've got your target matched in terms of what your market is, what your soil is, what your climate is, you can come up with the ideal idiotype for that. And then if you know what the functions of the genes are, you can produce an idiogenome, that is a whole genome sequence that you think is going to produce that ideal phenotype. And of course, I say you think it's going to produce that ideal phenotype. That's where we're never going to get rid of evaluation and phenotyping, because having produced your ideal genome, you're then going to have to phenotype it in order to confirm that it does actually produce the kind of variety that you predicted. And if it does, great. If it doesn't, you then have to refine your genome to phenotype relationships algorithms, which is a step forwards. So yeah, it's coming up with the idiotype first and then working backwards towards a genome sequence that you think will produce that idiotype.
Thank you. Thank you, Rory. We've come to the end of this Grow webinar. Enjoyed it very much. I hope a lot of the other people also did. Thank you so much for putting in the time and for showing up and trying to teach us also and creating a vision of where we should go. To all the people in the Crop Trust, thank you so much for your support today in managing the webinar. And all of you out there, it was great seeing you. Thank you for attending. We hope to see you next month. Bye-bye, everybody.
Thank you, and everyone else.